Have you ever just stopped and wondered how well, how it builds itself? You know, how does a single cell know it's supposed to be a heart cell or a neuron or even part of a leaf? Well, it all comes down to one of the most elegant and frankly beautiful processes in all of biology. In this explainer, we are going to unpack that exact process, the incredible journey from a master blueprint to a fully functional building. So let's dive right in and follow the instructions, from code all the way to creation. So this is the big question, right? Tucked away inside pretty much every single one of your cells is this set of instructions, and it's written in a language that only has 4 letters. So how in the world does that incredibly simple code direct the construction of something as complex as you, or a giant redwood tree, or even a tiny little bacterium? That's exactly what we're here to figure out. Okay, so our journey starts in the most protected part of the cell, with the master plan itself. And listen, this isn't some rough sketch or a first draft. We're talking about the meticulously detailed master blueprint for absolutely everything the cell will ever need to do or build. And its name? DNA. Right, so DNA, or its full name, deoxyribonucleic acid, is our master blueprint. The best way to think about it is like a super secure library, holding the original, priceless copies of every single instruction manual. It's built to be incredibly stable and heavily protected, making sure that information stays perfectly intact for the entire life of the organism. What's really wild is just how simple the language of DNA is. It's got just 4 chemical letters, A, T, C, and G. But here's the magic. They follow an incredibly strict set of rules. Adenine, or A, will only, and I mean only, ever pair up with thymine, T. And guanine, G, will only ever pair up with cytosine, C. That's it. This strict pairing means you always have a perfect 1-to-1 ratio between them, and this simple rule is the secret to how DNA can store and copy information so flawlessly. So, these 2 strands, with their letters all paired up perfectly, don't just lay there flat. Nope, they twist around each other to form that shape we all know and love, the double helix. And this is just a brilliant example of how form follows function in biology. This elegant twist isn't just for looks. It compacts the molecule, and, most importantly, it protects that precious sequence of letters, the actual genetic code, which gets tucked safely away on the inside. Okay, so we've got the master plan. But how do we get those instructions to the construction site? I mean, you would never take the one-of-a-kind, priceless master blueprint out to a muddy, chaotic building site, would you? Of course not. You'd make a cheap, disposable, working copy. And that is exactly what the cell does. This whole process of making a copy is called transcription. The cell carefully unwinds just a tiny section of the DNA, a single gene, and creates a temporary message from it. This messenger molecule is called RNA. It's basically a short, single-stranded memo designed to carry the instructions out of that protected library and over to the cell's factory floor. Now, what's really fascinating is that not all life handles this the same way. In complex cells, like ours, we're eukaryotes, the process is super precise. One gene gets transcribed to make one RNA message, which in turn is used to build one specific protein. Very specific. But in simpler organisms, like bacteria, prokaryotes, it's all about speed and efficiency. They can grab a whole group of related genes and transcribe them into a single RNA message, letting them produce all the proteins for a specific job all at once. It's just a fantastic example of different evolutionary strategies, precision versus pure raw efficiency. So, our little RNA messenger has left the library with its instructions. But where is it headed? It's on its way to the construction site, the factory, where the actual building is going to happen. And in the cell, this factory is a tiny but incredibly powerful machine called the ribosome. And the building process itself has a name, translation. Because this is literally where the language of the blueprint, that code of nucleic acids, gets translated into the language of building materials. The ribosome clamps onto that RNA message and starts reading the instructions, code by code. Now, the crucial part here is how the ribosome reads the message. It doesn't read one letter at a time. No, it reads the RNA in 3-letter words. Each little group of 3, called a codon, is a specific instruction that basically says, add this specific building block right here. It's kind of like a recipe where every 3 letters tells you exactly which ingredient to add next to your growing chain. So now the ribosome has built this long, kind of floppy chain of building blocks. But our story is not over. Not by a long shot.
In fact, the most magical part is just getting started. See, a simple chain isn't a machine. To actually do anything, it has to fold itself into a very precise three-dimensional shape.
And this, right here, is the absolute key to everything. That specific order of the building blocks, the amino acid sequence, is what dictates exactly how that chain is going to fold. It's this incredible act of self-assembly, where the one-dimensional information in the sequence just spontaneously creates a complex three-dimensional structure. That final shape is what gives the protein its power. Now, this folding happens in a few stages. First, you've got the primary structure. That's just the raw, linear sequence of the chain. Then, little parts of that chain start to form simple repeating shapes like coils, called helices, and flat sections called sheets. That's the secondary structure. After that, the entire chain folds up on itself, creating a totally unique and complex 3D object, the tertiary structure. And for a lot of proteins, there's one final step, the quaternary structure, where several of these already folded chains come together to form one giant functional machine. So why does this shape matter so much? Because in biology, structure is function. It's everything. For an enzyme, this precise folding creates a little pocket called an active site, which is perfectly shaped to grab onto another molecule and kickstart a chemical reaction. For a structural protein, that shape is what provides strength and rigidity. The job a protein does is completely, 100% dependent on its final folded shape. All right, let's just take a quick step back and look at this entire process we've just walked through. When you see it all together, from that protected master blueprint all the way to the final working machine, it's a truly elegant and amazing flow of information. So let's trace that flow of information just one last time. It all starts with the DNA, our permanent blueprint. That blueprint is used to make a temporary copy in a process called transcription, creating the RNA messenger. That messenger then travels to the ribosome, our factory, where it's read during translation. The ribosome builds a protein chain, which finally folds into a complex 3D machine that goes off to do a very specific job in the cell. Blueprint to copy, to construction, to the final working product. It's beautiful. This entire process, this beautiful flow from DNA to RNA to protein, is what biologists call the central dogma. You can think of it as the fundamental operating system for every single living thing on this planet. It's elegant, it's efficient, and it is almost always perfect. Which leaves us with one final and really crucial question. What happens when it's not perfect? What happens when there's just one tiny typo in that master blueprint?